Okay, yay. It is working. Now it seems like it's a bit too bright in here, right? So let me turn off these lights here. All right. Don't fall asleep on me now. Okay. I also just started that iClicker thing, so if you want to get your iClickers out. And I forgot to mention that you do get just being alive in class participation points. Okay, so I did put in the option to count just what is it? It's not absence, it's like presence, okay? So if you're in class, you're gonna get a point for presence. You try to answer questions and you get point for participation. If you answer them correctly, then you get 2 points. It gets better. I hope it's not too dark in here now. Okay, so I have a lot to cover today, and hopefully this thing is working too. Yay, everything works today. It's a good day. All right. So we left last time just having a brief overview of the job for the immune system and the components of the immune system, and this idea that you have microorganisms everywhere, right? And you have this microbiome, and bugs have usually a bad rap, right? Just because some bad bugs cause diseases, and yeah, that's reality. But a lot of the bugs you never hear about, those are the good bugs. And I want to mention one more thing before we really start getting into the mechanisms and cells and everything else that has to do with the immune system. The bugs that you're first exposed to, like for example, at birth, right? Those are also very, very important, because not only they're going to help to shape the immune system of the baby, of the newborn, but they are also going to determine absence of some diseases. Like for example, babies that are born through C-sections, they tend to have more allergies than babies that were born naturally, okay? And also, babies who are growing up in an environment that is extremely clean have more allergies than babies that are in a normal environment, right? And I can tell you stories, if you're, I'm assuming that nobody's here is really a parent, or, maybe don't have like small babies in the house, right? But I can tell you, like, by the time you have your first child, oh my God, you go obsessed with cleanliness, right? Everything has to be sterile around the house, right? And that was, yeah, me with my first baby. Like, oh my God, everything has to be cleaned and totally, totally spotless. And yes, my baby was born through C-section, not by choice, but due to emergency of preeclampsia, yeah. I could tell you some stories about that experience, but that goes away from the physical aspect of the class, when you step on the other side of life or death and some kind of thing. But anyway, this is an interesting story, but not for now. Anyway, so he was born through C-section, and I was so fanatic about keeping everything clean. So he had asthma, he had severe allergies, he had hypersensitivity, you name it. All right, so by the time my second baby was born, I became a little, lazy or tired, So I had to clean some stuff. Oh well, it's clean enough, Oh, I got some emergency. It's not bleeding, no broken bones. It's okay. All right. The second child is usually a lot tougher than the first child, right? He never had allergies. I'm not saying don't keep your house filthy, but don't go too crazy, okay? Because you need some exposure to the environmental microbiome, right? In order to develop your immune system properly. Okay, so that goes, with that talk. Okay, so here, some terms that you have to know. Okay, so a pathogen, that has to do with the bed bugs, of course. Pathogens will cause disease. So that's the definition of pathogens. And what kind of pathogens you have out there? Well, you have one of these 4 categories here. Of course, viruses, bacteria, some fungi, some parasites. Okay, all right. So the way you study pathogens, that is called, of course, pathogenesis. Okay. Now, the way that your immune system will attack and get rid of the pathogen, that really has to do with the nature of the pathogen. So if that pathogen is living outside of the cell, so that is extracellular, the immune system is going to behave a certain way. If the pathogen lives inside of the cell, then your immune system is going to behave in a different way. So it is tailored to attacking the pathogen where the pathogen likes to work. Okay? All right. So the term immunity is really meaning away from burden or free from burden. So this is really the definition of that word, or the root of that word. Okay. Okay, so let me see here. Yeah, of course, pathogens have certain triggers for the immune system that is going to activate in one way or another. Okay. Now, interestingly, if you look at this here, most of the pathogens prefer not to kill their host, because if you kill the host, then you have nothing else to infect, right? So most of the pathogens, they like to just, cause some disease, but not enough disease that the host will die. Otherwise, they have to find another host, right? So a lot of them sometimes just hang around and don't kill you, right? They need you, Okay. And a lot of times they even allow you to recover from the disease. And that is really at their species' expense, because if some of their cousins or very closely related pathogens decide to infect you, you're going to have some kind of immunity against that kind of bug. Okay. So most pathogens, or some here, but most of them are quite opportunistic. Okay. And now you have to also have this idea that if a lot of people like here, like you are in the same room, right, and we're exposed to the same kind of bugs, some people have different responses to the pathogen in comparison to other people. So some people have a more active immune system, some other people have a less active immune system. So the way that your system is going to respond is really going to depend on how your immune system works. Okay? So some opportunistic bugs are really going to take advantage of someone who has a weak immune system. Okay. So here I have a similar slide that I showed before, that it has certain viruses that cause disease. So here are just some examples of diseases that are caused by viruses, right? And we are all very, very much familiar with the COVID-19 type of disease, right? Yeah. And then you have bacteria here, and all different types of bacteria that causes disease. Then you have some fungi, and the most common one is really Candida albicans, right? That causes thrush as well as yeast infections. Then you have certain parasites that can cause a lot of diseases as well. All right, so here just looking at the whole, the big picture here of your immune system, right? So we say here the immune system is really a double-edged sword, because at the same time that it protects you against infections, and it can learn to perfect that protection, right? At the same time, it can cause certain types of diseases. Okay, if the immune system is not controlled, is not really self-controlled, then you can develop certain types of autoimmune diseases that can be very detrimental. So you have to have this very precise balance between attacking what is not good for you, but at the same time distinguish that from your own cells and not attacking your own system. Okay. Now, another good thing about the immune system, besides attacking invaders or pathogens, is that your immune system is very much in charge of getting rid of damaged cells. Okay, and mainly that's the job of your innate immune system. It gets rid of damaged cells by recognizing patterns that are strange in these damaged cells. So those damaged cells are going to have certain proteins that are, of course, damaged, and certain telltale signs that we call them damage-associated patterns. Okay, so your immune system is going to recognize those and say, oh, this cell it's not good. I think it is it's a cancer cell. I think it can cause trouble for the host. So then your immune system is going to go there and is going to kill that cancer cell. So this is very important to keep your body free from this type of transformed cells. Okay? And once we started to understand these mechanisms, that really opened up a whole field of immunotherapy against certain types of cancers. Okay, and nowadays we have in like a mainstream therapeutics. You okay? You have a lot of these new drugs that are intended to work with the immune system to kill these certain types of cancer cells. Okay. Right. So here we're starting with the lines of defense and their functions. All right. So we have to keep in mind here that innate immune system is the system that you're born with. Not only that you're born with, but is ready to work as soon as you are born. Okay? Because really you're born with both of them. But one of them has to be further developed, but the other one is ready to work. Okay? So the innate immune system has certain cells that are from your myeloid lineage. And those cells, remember, they have certain molecules that are genetically determined, right? It comes from the genes that are already there. And they can attack the invader always the same way. Okay, so it uses same strategies every time it sees some pathogen trying to get into your system. Okay. Now, besides that, the innate immune system, as we're going to see, it has several layers. First thing that you have to think about, what is protecting you against outside invaders. The first thing that should come to your mind is really like your skin, right? The skin is protecting the outside world from the inside world. Not only your skin, but also your mucosal tissue that is separating what is outside from what is inside. So all this physical barrier that is considered as your first line of defense, or also part of your innate immune system. But besides that, you also have to think at the molecular level, okay? Because you are protected from some pathogens just because the pathogen doesn't have a way to infect your cells, okay? So here, this is why I mentioned here, the mechanism of innate immune system protection has to do also with the lack of receptors that certain organisms would need in order to invade your cells. And here we have two examples that will go into a little bit more details, right, on how that works. So this is all part of this innate thing that you're born with, and the thing is ready to work. You don't have to do anything else for this thing to work, okay? When you compare that to your adaptive immune system, so this is really like the third line of defense of your immune system, okay? So the adaptive immune system, it cannot attack the pathogen right away. It has to learn what is the pathogen, and then it has to develop certain weapons against that pathogen. And then it needs to be activated to start working. So your adaptive immune system is really going to start working only after about a week or 10 days after that initial exposure to the pathogen. And not only it takes time to learn, but it's also dependent on the function of innate immune system cells in order for that thing to learn. Okay, because without the innate cells recognizing the pathogen, then the adaptive system is never going to learn. Okay, it's not going to learn on its own. All right. Now, as we're going to see, some examples of how you're going to develop this this adaptive system, we have to keep in mind that mainly you can develop it in two ways. Either you see the bug directly, or you receive weapons from somebody else who has seen the bug. Okay, we're going to go into a lot more details on that. So here is this example of innate immune system that really protects you from getting this particular type of disease that is caused by the virus. Okay, so there is this virus that is called canine distemper virus. okay? That if you get exposed to the virus, you're not going to get infected, okay? Why? Well, because our cells, they don't really have the receptor that can bind to the virus. So when the virus gets close to our cells, the virus does not recognize the receptor. It's not a part of the tropism of the virus, right? So then the virus is like, oh, well, I can't penetrate that cell. I might as well just go look for a different host. So we are naturally immune to that type of infection. We didn't have to learn anything. We just didn't open the door, okay? Now, this canine distemper virus, when it finds the species that has the proper protein on the cell surface, like species, for example, dogs or raccoons or skunk, okay? So this virus will be able to penetrate the cell and get that host quite sick. Actually, it's usually fatal in wild animals that have those receptors, okay? And that's why we have vaccines for our pets, right? Okay. Now, this is interesting. Although we are protected against infection from the virus, if you get in contact with an animal that has the virus, and then you go and you interact with an animal that was not exposed to the virus before, you can serve as the carrier of the virus and then expose the other animal, okay? So although you don't get infected, but you might be able to just transmit the virus to some of these species that are vulnerable. Okay, we have to keep that in mind too, because a lot of viruses they are species specific. Okay, and we see now, in the news we started having problems with the bird flu, right? And we see that some products like raw milk are contaminated with some bird flu. And I think I heard yesterday or the day before there was one person that died from being infected with the bird flu. So some viruses are able to cross species and mutate very quickly, especially when you talk about influenza viruses. They're very good at mutating, right? But then some other viruses, they just don't mutate, and this is why we can have effective vaccines against those viruses. And again, we're going to talk a lot more about mechanisms of vaccinations later. All right. And then we have this case here. Who heard of the CRISPR babies? Okay, I want to see like hands, like who heard of CRISPR babies? A few people. And here is a case, right, of these cute little twin girls. And what happened here is that this guy, like Junqui, right? He was actually a bioscientist, a biophysicist that thought about developing this immunity against HIV infection. Okay, so he came to the conclusion, or he was coming to the idea that HIV, when it infects a cell, it needs a receptor. So that's your CD4 receptor, right? And as we're going to see later, CD4 is a main receptor for adaptive immune cells, right? So the T cells, like T helper cells, right? Now, in order for the virus to successfully infect a cell, not only it needs the receptor, but it also needs a co-receptor. So this guy knocked down the co-receptor for HIV, and that way it really limited the ability of the virus to penetrate the cell that it wants to infect. Okay, and this whole idea was that he wanted to solve a problem of having babies, right? Fertility problems in couples that, for example, the father is HIV positive, but the mother is not. So he was thinking that if he's able to block the entrance of the virus for the fetus, then the fetus is not going to get infected with HIV. Okay, and as far as I know, everything went fine, and as you can see with the babies, the experiment worked. And last time I checked, the babies, the children were doing very well. Okay, Also, I don't know what happens to the guy. I haven't, looked recently, but last time I checked, he had conveniently disappeared. You know what I mean? Wow. Oh, okay. Not much said on that. A lot of controversy with those types of CRISPR babies, right? Who has watched the movie Gattaca? One person who has heard about the movie Gattaca. Oh, you gotta watch it. You have to watch it. Okay, I don't know how to spell it. Okay, I'll try my best here. Okay, so it's like G-A, something like this. Of course, it has to do with nucleotides in your DNA, right? I think it was from the 90s. It's an old movie, okay? And there is one scene in that movie that I have no idea why they put it upside down. We kind of have to go like this and watch it. Oh, well. Watch the movie. You'll like it. It's not part of the class, but just for entertainment for a Saturday night or something like that. Okay. All right, so let's delete Gattaca here. All right. So here, innate type of response. Okay, the innate system is always going to respond the same way every time it finds a pathogen. Okay, and one characteristic or a hallmark of innate immune system is development of inflammation. Okay, so let's keep that in mind. Inflammation is part of your innate immune system strategies. Okay, now this system has to work very quickly, and because of inflammation, it is capable of increasing your body temperature and causing fevers. Okay. At the same time, of course, it acts always the same way every time it encounters a pathogen. So we say, for all practical terms, that it does not keep a memory of who it has seen before. We know nowadays that that's not 100% true, okay? But for purposes of mechanisms that we're going to study in this class, we just say that it does not have a memory, okay? All right. Now, how is it that the system works? First, we have the physical barriers. Then we have your chemical barriers, okay? So when you think about physical barriers, you think about skin and you think about mucus, so skin and guts, okay? At the same time, you put that your reproductive system, right? Because that is a very good site of entry for something that is outside to get into your system, okay? So skin and mucous membranes, not just guts, right? Besides that, you also have some structures in your mucous membranes that have these specialized hairs that we call cilia, and those are also physical barriers that they help to kick out the microorganism before it can penetrate some cells, right? So first thing that you're going to find in the innate immunity is a physical barrier. Okay, it's right there. Okay, it was working too now. Okay, I should not fight with it. Okay, physical barrier. Okay, and then you have your chemical barriers. All right. Now, if I go back in the PowerPoint, and then it doesn't work for the recording, so I'll just go along. All right. So first thing you have in mind, you just think about what is it that a pathogen has to do in order to cause a disease. First thing, it has to pass from the outside to the inside. So that's your physical barrier. Second thing, it has to survive whatever you have in that physical barrier, which is related to chemical barrier. You have enzymes, okay, that are intended to kill the pathogen. Not only that, but you have certain enzymes intended to kill parts of components that make up pathogens, okay? Like, for example, in your fingers, you have these enzymes that are called RNases. Okay. Anybody work with extracting RNA here in the lab? No? Anybody work in the lab, like, at all? One person. Okay. Did you extract the DNA? No RNA, no DNA? Oh, you missed the fun part. Okay, DNA, piece of cake. You have little kids from like the big companies, QIAGEN, blah, blah. You spin a few times. At the end of the day, you have your little pellet. Everybody's happy, except if you suck out the pellet when you're trying to dry it out, and then you have to start over, for 8 hours the next day. But anyway, easy. It's easy to extract DNA. When you're going to isolate RNA, it's a pain. It's a pain in the neck. Why? Everything has to be very, very clean. You absolutely have to wear gloves that are very clean. Okay? Why? Because in the tip of your fingers, you have these enzymes. They're called RNases. And if you touch anything, especially if you're trying to isolate RNA, those enzymes are going to kill the RNA. So you're not going to isolate anything because everything is going to be degraded. Pain in the neck. Big pain in the neck. Okay? But that's one form of innate immune system defense. Because you can imagine some viruses have RNA as their genetic material. So if you have RNases on your fingers, you are going to kill the virus and you're not going to get infected, right? So you have the physical barrier, the skin, you have mucus that is also physical barrier, and you have chemicals that are intended to kill whatever is trying to get in you. And then besides that, you're going to have those innate immune cells that always act the same way, okay? Now, when you look at innate immune system, there is a lot that we don't know about it because we can only study what we can have as a model in the lab, okay? And a lot of times we don't even know what the immune system or the innate immune system is doing because we don't really get sick with these pathogens they're trying to get in our system, okay? So the innate system, the innate cells are doing the job and nothing ever happens. So what you're going to study? Like, how are we going to study something that didn't happen? You know what I mean? It's kind of tricky to study the innate immune system. And then you try to put it in a lab setting, and then you, of course, take the cells out of their natural habitat inside somebody's body. You put in a petri dish, you put in a dish at all, and you're already activating them. So that's another problem. How can you get them not activated and study them in a lab? All right, so there are a lot of things that we still don't know about innate immune cells functions. Okay, but now let's say that your innate immune system did not take care of the pathogen. Now you are going to start activating your adaptive immune system, but only after your innate already did everything that it possibly could, okay, which was going through 3 different levels. Okay, physical, chemical, innate cells. Now you're going to start activating your adaptive system. And this is a part that is interesting here because your adaptive system, you are born with cells, right? But those cells are not ready to go to work. Okay, so you have some cells, but if you don't teach those cells how to work, then they are useless. And when your innate immune system starts teaching your adaptive immune system, now those cells start to transform from being totally naive. You call them naive, okay? From being totally naive to these cell types that are called effector cells. Those are the guys that are going to be helpful, okay? So it's not to say that you're not born with adaptive system. That's not true. You are born with the cells, but they are useless unless you teach them. After you teach them, they become effector cells. Now they come into action, okay? So when you say effector cells are not pre-existing, that means the naive cells are not the same as the effector cells. They're there, but they have a lot to learn, okay? All right. Now, every one of these effector cells, those are very, very specific for whatever they have to attack. Okay, so they call them antigen-specific cells. And what are the cells that do this job? They are your T cells and your B cells. Now you're going to see all different types of T cells, all different types of B cells, but those are the cells of your adaptive immune system. Okay, and they are of the lymphoid lineage. All right. Now, those cells are really cool because those are the guys that do develop a memory of what they have seen before, okay? Now, again, in order to develop the memory, first thing that has to happen, it has to encounter, yeah?
How long does the immunological memory last? Is that your whole life?
Depends on the pathogen, because some pathogens, you do have memory for your whole life, but some other ones, like depending on like the type of bacteria, for example, against some viruses you do have for your whole life, but depending on the type, if the virus doesn't mutate or it does, and especially when you have immunity against certain bacteria, that is a bit trickier because you can lose the immunity after a while. Some memory cells, they don't hang around forever, and that's why you need booster shots. Yeah, it really depends on the pathogen. Yeah. Right. No, with COVID-19, that's your coronavirus, and that's RNA virus, and they do mutate a lot. Now, the thing with the COVID-19 is unique because not only you had to have a virus that we were not exposed to before, but the virus also had that virulence that was very pathogenic. So it was this combination that if you look throughout history, every 100 years or so, you're gonna find some virus or some bacteria that shows those specific types of characteristics that you have to have most of the population vulnerable. The coronavirus is not new, it just didn't show up 2019. Now this virus was really nasty, and sorry to say this, but I do believe that it was a laboratory accident. I want to think and believe that it was an accident. Otherwise, I'll lose faith in the human species. So yeah, but it was designed to be very pathogenic. So that's yeah. And now after everybody got exposed to it, a lot of people, of course, developed immunity if they survived, right? But the virulence of the virus goes down as the passages increase. Like after it's out in the population for quite some time, the virulence tends to go down and down. Okay, because coronaviruses, we know it causes flu-like symptoms. I mean, we know this forever. But like this one, that was very, very bad. But yeah, we have immunity against the most severe type of coronavirus, like COVID-19. But then the other viruses that keep on showing up, they have less and less virulence. So don't worry too much about them. But yeah, it really depends on first of all, of course, in your own immune system reaction, but also in the virulence, the virus virulence. Okay. So let's see here. So your adaptive response, first thing that has to happen, it has to be primed. Okay, it has to learn exposure to that particular pathogen. Okay, it will learn either directly, like getting infected with the virus or the bacteria, or it can learn by artificial exposure. So those are your vaccines. Okay, but it has to be primed. If you don't prime it, it's not going to work. It's not going to develop. Okay. Advantage is that it's going to have the memory. Okay. Disadvantage is that it's not fast. It's going to take at least 10 days for it to start working. Okay. So advantage again, that is it has a memory. All right. All right, so what is this memory thing? And yes, I did rearrange some of those slides to make more sense as I was talking it, as I have to talk it throughout the class, okay? All right, so here, let's see what the memory thing is about. So here, when you look at this light blue graph, okay, this is representing the response of your innate immune system, okay? So here we say at day 0, this is when you're exposed to, let's say, the virus, okay? So right away, your innate immune system mounts up a response. And that means inflammation, and all these cells are going to the site of infection, and they're really trying very hard to kill the pathogen. And let's say that if you kill the pathogen, then everybody's happy, you did not get sick, and everything is cool. All right, but now look at this. After around 10 days of pathogen exposure, this is when you start to see a response of your adaptive immune system. So by the time you hit 2 weeks, your adaptive immune system is really at work, if you still have some of those viruses that need to be killed, okay? If the innate immune system totally gets rid of the virus, then you don't worry about it. Then adaptive immune system is going to have a memory, but it is not going to be activated. Okay, so let's see here. Day 0, innate immune system went to work, had certain response, and then it went down because you don't want it always activated, otherwise you can have problems, right? So it has to be down-regulated, everything goes back to normal. Now, let's say here, after a month or so that you totally recovered, let's say that now you got in contact again with the same pathogen. So here is another day 0 after 30 days of first exposure. So what's the first thing that's going to happen? Your innate immune system is going to respond exactly the same way as it did before. It totally doesn't remember that it has seen that pathogen before. Okay? But now, at this second exposure, you're going to have immediate activation of your adaptive immune system. Okay? At the same time, look, same time that you get your innate immune system activated, you are going to get your adaptive system activated with the second exposure. Now the amount of activation is huge for the second exposure for your adaptive immune system, because it really is going to remember that it has seen that guy before and it has the proper weapons to attack the guy. Okay, so the level of this response is a lot bigger and it's a lot stronger than the first exposure. So that's what we mean by memory of who has seen, of what he has seen before. Okay, the secondary exposure is a lot bigger. Now, keep in mind that this type of reactions are not only related to pathogens, but when things go wrong, it's also related to allergies. Okay? Now, let's say that if you're allergic to something and you have never exposure, you never came in contact with that thing before, the first time that you get in contact with that thing that is not a pathogen, is just, let's say, pollen. Okay, first time you get in contact with the pollen, you're going to have some inflammation, some stuffy nose, your innate immune system is going to go into action, but then your adaptive immune system is going to learn that pollen is not good for you, but you're not going to have a huge response first time you see it. Okay, second time you see pollen, oh, that's when you get those bad allergies, right? Because now your adaptive system is going to think, we got to kill pollen, right? It's not very harmful, but it doesn't know that, right? So your body is programmed in a certain way to react to certain things. And when those things are harmless, that's when you have allergic problems, okay? Just keep that in mind. Okay. All right, so let's start looking here at characteristics of some of those cells that are very much important, in this case here for your innate immune system. Okay, you have here your monocyte. Okay, so you have myeloid lineage cells. We're going to look more again at those later. You have here neutrophils, and those like to really like float around in the blood. Okay. Now, keep in mind that these innate cells, they like to sit in the tissues and keep guard of those tissues. So you have, for example, the cell that I don't have here, but you have, for example, cells that are macrophages right under your skin, okay? And those used to be called Langerhans cells. Now we just call them macrophages, okay? Now, they are sitting there and just waiting for somebody to try to come across, and then they're going to attack. Okay, so you have tissue-resident macrophages. That's part of your innate immune system. But then those cells are sitting in tissues. Besides those cells, you also have cells that are always patrolling the blood and the lymph. Okay, so all their specialized cells, and those are your neutrophils. Okay, so we have a lot more cells we're going to look at now, just as a big picture. So now when you look at cells of your adaptive system, you have B and T cells. Look at the B and T cell as a lymphocyte. You see that most of the cell is just like a big nucleus, right? It has very little cytoplasm. And those cells, you can't really distinguish if it is naive or effector cell but you know that they all kind of look like this, and they're floating around in the in circulation, or they're mainly part of your lymphatic circulation. All right. So you have different cells that have different functions. Now, when these cells get activated, they start to divide very, very quickly because they know that they need to attack a certain type of pathogen, right? So you're going to need a lot of them that are specific for that pathogen to be activated. So this is what we call like clonal expansion, as we're going to see more details later as well. Okay. All right, so again, a big picture of your innate system. You have your physical barriers, you have your chemical barriers, and then you have your cells. Your cells are going to have certain pattern recognition receptors, right? And those are the ones that are coded by the DNA, and the DNA code doesn't change, or that's why they're innate, they're all there, okay? And you have a certain number of these receptors that's not really a lot. It's in the hundreds, okay? Besides your cells, you're also going to have free-floating proteins that are part of your innate system. And those proteins are very important for functioning of your complement system, okay? So those proteins are made in the liver, and they're put in the circulation, and they go around looking for pathogens. Okay, they're not cells, they're just proteins. Once they bind to pathogens, to some patterns in the pathogens, once they bind to the pathogen, then they're going to trigger this whole reaction and call on different cells and try to destroy the pathogen. So important for the innate immunity is not only the cells and pattern recognition receptors, but also your complement system. And again, your cells, the effector cells of your innate system is really the ones that eat up the invader, so we call them phagocytes or phagocytic cells. So when you look at your adaptive system, you have two cell types to remember, B and T, that's it, okay? Oh, by the way, also for the innate system, you also have those natural killer cells, okay? And those are lymphoid lineage. We're going to look at them later again. Okay, so adaptive system, B cells and T cells. They have to be taught, right? You're not born with effector cells. You're born with all naive cells. They don't know what to do. Then once you teach them, they become effector, then they come into action. And they're highly, highly specific. All right. So here, a little chart of comparison between your innate and adaptive system. One thing that you really, really have to get the concept of is how quickly they are working. Your innate system is very fast because it's the first thing that is going to protect you, right? So it's going to jump into action like right away. You're talking minutes or hours, all right? Your adaptive system, that's going to take days. First exposure, it's going to take days. It's going to take a long time. Now, how specific are they? This guy is not a lot specific, okay? Comparison to adaptive system, it's highly, highly specific, right? Now, how are they going to respond? This guy is going to respond the same way every single time, but your adaptive system is going to respond in a particular way depending on the pathogen. Okay. Major components, again, easy to think about innate system, whatever is already there, right? Major components of adaptive, T and B cells. Okay. Now this other chart here is also saying, about how fast they are. This is innate system is always there. Okay, recognition of patterns. And here I added this table here because I also put the cells here. Okay, the cells that are important for those systems. So innate system, you have to think myeloid cells, natural killer cells, and all the other physical and chemical barriers. Okay, then adaptive system, of course, you think about your B and T cells. Oh yeah, antibodies is included there because you can have free-floating antibodies, and that's like a protein that is floating in the blood, okay? But antibodies is really the blood component that is not a cell of your adaptive system. All right, so this is just to emphasize things that I have already mentioned, so you can read that on your own. Okay. Right, let's see here. All right, so this is a big picture of how the systems are working. Okay. First thing that has to happen, you have to be exposed to the pathogen. The pathogen has to go through that physical barrier. If it passes the skin, okay, or the mucus, or if you have a cut and it goes through the cut, right, point of entry here, if it passes the skin, now you're gonna deal with chemical barriers also in the skin, chemical barriers, but now you start getting your cellular components here, okay? So your cells, innate cells, are going to detect this, is going to recruit other cells that are innate, is going to start inflammation, and some of those cells are going to carry the pathogen to a secondary lymphoid tissue, and it's over there that is going to get in contact with your T and B cells. Okay? When this guy, the innate immune system, goes over there and teaches your B and T cells that there is an invader, now you're going to start mounting your adaptive system response. Once you have effector cells, those are going to get into the circulation. That's why you have a little heart here. Okay, get into your blood and go to the site of infection and help your innate cells to get rid of the pathogen. Okay, so we have several things that are going on here. But without your innate cells, you don't have activation of your adaptive cells. Okay, so this is going to talk to each other.
Question.
Yeah.
What's an example of a primary lymphoid organ? Bone marrow?
Yeah, there are only two primary lymphoid organs. The first one is your bone marrow, that's your red bone marrow, and the second one is your thymus. Only these two. Now, your lymph nodes and gut associated and everything else that is not bone marrow or thymus, those are all secondary. And so you talk about secondary mainly where your B and T cells develop. Okay, your primary cells, your innate cells, they do all their development inside of the bone marrow. Okay, they come out of the bone marrow ready to work. Okay, only your T cells and B cells that are going to need, well, T cells are going to need thymus development. So that's also why the thymus is called primary. Okay, and then your B cells and also T cells are going to need your secondary lymphoid tissues to further develop. Okay, and yes, we're going to go to a lot more detail because, yeah, this is only the big overview of the whole thing of the immune system. And I'm sorry if I'm causing a bit of a headache, but I'm trying to put it as simple as possible in a way that it will make a flow sense, okay, from trying to invade the host and what are the barriers and what are the things that are getting activated. And we're going to get into a lot more details and all the stuff that is happening. Okay. All right. So first thing here that you really have to start paying attention, a lot of attention to, right, is classification and development of immunity. Okay. If you look here, okay, immunity is branched into two, right? So you look here, your innate immunity is not much happening over there. Innate immunity, you already know your innate cells, and that's how they work all the time. They don't learn anything new for here, okay? And they're always working the same way, everything is cool. When you look at your adaptive immunity, now things start to get more sophisticated, okay? Why? Well, you can have adaptive immunity that you get naturally, or you can have adaptive immunity that you develop artificially. So let's look at natural immunity first. If you receive antibodies that are ready to work from somebody else, that is called passive natural immunity. There are only two ways of getting these antibodies. Either you are breastfed or you get serum from somebody who already has developed the immunity. Okay? So passive, it can be maternal, natural, or passive can be you receive from somebody. All right, so let's stick to the natural. How are you going to get natural passive immunity? Only if you get breastfed. Natural. naturally, okay? So natural immunity with antibodies that are already made, breastfeeding. Now, natural immunity with the culprit of the disease, that is called active natural immunity, and that means you got the infection yourself, okay? Now, what would be an example here? Let's say that, you know, the weather is colder, and the flu virus, it likes to replicate in, you know, inside of your lungs if the air is a bit cooler, right? So you go out and you get the flu. That is a natural active type of infection that you got, and you develop secondary immunity because you were exposed directly to the virus. Simple, right? Okay. Now let's see artificial ways of developing adaptive immunity, okay? You can get it passively, okay, if you get antibody transfer from someone who is already immune to that disease. Now, example here of COVID-19 treatment, right? They had antibody treatment in case a person was really sick and, you know, started developing the COVID symptoms. Then used to go to these treatment centers, and they would have serum antibodies, and they stick it in your veins. So those are the antibodies that are ready to attack the virus, but somebody else made them, okay? So this is passive artificial immunity. It's not going to last for a long time. It's only going to last until those proteins that are the antibodies, until they get degraded in your body, okay? So artificial passive, you got a serum from somebody else who made the antibody. Now, artificial active immunization development is immunization, okay? Because now you're not getting antibodies from somebody. You're being exposed to something that is not as bad as the original pathogen, but is going to protect you against that original pathogen, okay? So vaccines are artificial active type of immunization. Okay, and we're going to look into more examples and more circumstances of how that works. All right, so active versus passive immunity. When you look at passive, you're thinking about treatment with serum from the blood that already has antibodies, okay? You're thinking about breastfeeding, okay? And let me see here. Serum, yeah, this is about the same, okay? Passive immunity, if somebody does not develop their own antibodies, they have to get it from somebody else. It's all passive. When you think about active immunity, you are thinking either about vaccination or full-blown infection with the pathogen itself, all right? Vaccination or infection. Oh, finally, your first question. Let's try this out. Right, vaccination is a form of I had to put that option C over there just for fun. Please not anyone answer C. Does anybody need more time? Can I cut it short? I will let the whole minute go. Okay, so what's your answer? It is active. Okay, it is active immunity. I should say artificial, right? But it's active. Okay. Passive, you have to think antibodies, right? Immunization, you think active. All right. And the government conspiracy is just that I wanted a third choice for that, right? I really don't believe that anything to do with vaccines is a conspiracy of, whatever. Okay, I'm not getting into that. This is a scientific class. Yes. Science should not depend on government affiliations. All right. Not very realistic, but let's pretend that it doesn't. All right. All right, so lines of defense. So here we see. First line, big barrier. Second line, smaller barrier. Third line, adaptive immune system. So I put it here so you can go back and look again into these different levels of defense so that it makes sense. And as you have seen, a lot of times I have some repetitive concepts just to make it easier for you to study when you go back and go through the slides. Yeah.
Does the pathogen always reach the adaptive layer?
Yes. Yes. And that's a good question, because sometimes you have certain pathogens that, although they have the same names, they are different pathogens because their genetic code changes. But your immune system has already seen another pathogen before it changes. So yeah, you always have the immunity. Yeah. Yeah.
So with the first line of defense, it's like the pathogen getting into your body, but second.
Yes. Yes. Actually, technically speaking, you have like 3 lines of defenses. So first is the physical barrier, and that you can include chemical barrier as well, like enzymes or or proteins in the fluids. Okay. Second line is innate cells, and then the third line is adaptive cells. Okay. All right. Okay, so here I included this slide here because I further broke it down into 2 components for your adaptive system. Okay? So you have your humoral component. That means it's stuff that is floating around in your blood or in the lymph. Okay? Humoral component of your adaptive immune system is really floating antibodies. Okay? You call it humoral because they say it was in the humors. They used to call the liquids like blood and lymph, they used to call those humors. Don't ask me why, right? Okay. So for your adaptive humoral, you have antibodies. Now, adaptive system, you have also cells that are going to work in there, right? So you call those cell-mediated type of immunity. Now, for your innate system, you also have a humoral component and a cellular component, okay? The humoral component of innate system, that is your complement system. 2 of them, not all of them. We're going to get into a lot more details in complement, okay? And then the cellular component are the cells of your innate immune system. All right, so here, again, levels of defense. You have those passive barriers, mainly skin and mucosal tissue. Okay. All right, so here, again, you have non-specific and the innate. Let me see what is important here. Oh, this is just repetitive, so we can read through it. And this is again repetitive. I might just take this out just so you can see. All right, so here a bit on the vaccination, right? So this is the artificial active type of immunization, okay? And we see here that going back to the history of vaccination, you have, for example, here the smallpox example, right? So when vaccines first started to be developed, Jenner, right, the doctor, the scientist, he noticed that while a lot of people were dying from smallpox, there was a specific population that was not contracting smallpox. Okay, so those were the ladies that used to milk the cows. You know the story? Have you heard that story before? Yeah. So ladies, their job was just to go, the milking ladies, they go there, they milk the cow, and they didn't get smallpox, right? So that guy Jenner, he associated something with milking the cows and not getting smallpox. So he decided to run an experiment with a human subject that would definitely not be approved in today's IRB standards, right? What did he do? He got this little boy that was not infected by smallpox. He really wanted to saved this boy's life. And he said, well, if the cow ladies are not getting infected, let me see what the cows have that the ladies got, and I give it to the boy. Of course, it makes sense, right? So he goes to the cows, and they see that the cows had a type of disease that was very similar to smallpox. It was not smallpox. It was actually cowpox, obviously, right? Cowpox, smallpox. It should be human pox, smallpox, big pox, and cowpox. All right. So he goes there and he bursts a little a blister from the cow. He gets the whatever pus and stuff he could get from that blister, and he goes to the boy and he makes little little cuts through dots through the skin, and he infects the boy with the whatever was causing the cowpox. Luckily, the boy did not die, right? The boy was fine. After some time, he decided to test his experiment. So he got smallpox virus from somebody who was actively infected with smallpox. And he did the same thing. He got a blister. He took the stuff out of the blister. He went to the boy. He infected the boy. And sure enough, the boy did not die. Had the boy died, we'd never have heard of this, right? So that was the first case of vaccination. Why is it vaccination? Because vaca is cow. So they got something from the cow. Let's call it vaccination. All right. I hope everybody has heard that story before. That's why I could fly through it, right? Imagine if the poor boy actually had died. Well, at the same time, I mean, you would never really have figured this out if somebody crazy like that guy wouldn't have done this kind of experiments, right? Talk about the double-edged sword of science, right? So that's the story with the smallpox. Now, the experiment that uses this type of exposure to a very similar virus that's not going to kill you, right? That is called variolation. Now, that very similar virus, it's not as strong as the one that kills you. So we call that type of pathogen, we call it attenuated, or, not as bad, okay? So most of vaccinations with live viruses are really using viruses that are attenuated. Some of them can get you a little sick, but hopefully not kill you, okay? So this is a type of attenuation, variolation, attenuation type of experiment. And here is that exposure, that experiment done by Jenner. I mean, look at the time. This is 1798. This is before the 1800s, right? I mean, it was impressive that the guy actually could have, thought about that stuff. Right? All right, then later on, we had other very important discoveries, and we do have the experiments done by Louis Pasteur, and that had to do with using an attenuated strain of bacteria that could cause cholera, and then the vaccination just took over, right? And took off, I would say. So we have certain diseases that were totally eliminated, like the smallpox, right? We don't see people getting sick with smallpox anymore. And actually, you do have some smallpox viruses that are only kept frozen in certain laboratories. You know where they have those viruses?
In
Huh? No. I don't think naturally. In some labs.
No, in
somewhere.
the lab.
Oh, I don't know about that. Really? Oh, okay. I thought it was just in Russia. Yeah. Yeah, I think, yeah, they have one in Moscow, and they have one in the CDC in Atlanta. And it's kind of scary to think that Russians keep those viruses too. But then they're going to say the same about us, right? Anyway. They do have those viruses frozen somewhere. Oh, well. All right. Which I had just flown before the previous slide that was talking about herd immunity. That's a concept that you have to know. Oh, if everybody's awake, that's the time that I give you the clue on, is it going to be on the test? Kind of clue. Okay. I tell you what I'm going to ask. You have to know about herd immunity. What is herd immunity? That's when enough of the population is vaccinated so that if someone is not vaccinated, they get protected because the pathogen is not going to be able to penetrate the population in general, okay? So herd immunity is like, even if I don't have the vaccine and everybody else does, how is the virus going to survive? Everybody else is vaccinated and, it's not going to get to me. That's the idea of herd immunity. And every pathogen has a characteristic number of people that have to be vaccinated in order for this herd immunity to happen. Again, when we get to the vaccines, we are going to know more about this, the mechanism. Okay, now we go to the question. And this is the time also that you can consult your neighbors and make sure that you get the right answer. Okay? So the question here is, which of the following statements actually describes herd immunity?
Oh.
Okay, we do have just a few seconds to go, and there is one person who gave me option A. So give this one person extra few seconds to change their minds. Because I really wanted to get all possible extra points in the class. They don't want to change their mind. Okay, I'm gonna stop it. Okay, but you have to change your mind to give me the right answer. Somebody changed their mind for the right answer. Okay, I'm gonna stop it. 1.5 minutes. What is the right answer? Oh, I think this person changed their mind in the very last second. That's a good E? Okay, your answer is D. Enough people are vaccinated and protected, so the ones that are not, they are protected as well. Critical number of people. All right. Other things that can make you sick or prevent people from getting sick, of course, clean water, okay? Cleanness, but don't go fanatic about it, right? Of course, your nutrition, right? Nutrition is very, very important for your immune system. Okay, so let's see here. Okay, this is self-explanatory. Oh, look, more points. I tell you, I felt very generous today. Okay. Antibiotics. What do antibiotics do? So this has to do with your prior knowledge on how these things work. Okay. Antibiotics are not antibodies. Antibiotics are some chemicals that you intake to kill something else. So I gave you half of the answer. Okay, so what's your answer here? Oh my goodness. Okay, I got a problem here. Because a lot of people answer B. Yes, antibiotics as we know them, mainly against bacteria, but yeah, you do have antibiotics against viruses. So this one, I'll just give you, freebie points. The correct answer is really D, okay? Yeah, you have antibiotics against viruses, and you have most antibiotics against bacteria. Yes, but it's also under antibiotics. Technically speaking, the most correct word is antiviral because viruses are not biologically alive, right? But in the general term, it's really antibiotic. So yeah, it's kind of tricky. So I'll just give everybody a point here. Okay. I will not fight this question. I'll take them out next time. All right. Oh, now you have here components, right? So we get into more details on, okay, let's pay attention here. Components of the immunity. You have cells, you have floating stuff, okay? That's it. All right. So, component of cell-mediated immunity. That is really something that has to do with your T cells. Because your T cells are going to do a lot of talk between cells and other cells to activate them, okay? So when you talk cell-mediated, think T cells. And we're going to study more of this later. Okay. All right, now we get into your primary and secondary immune tissues, okay? Primary and secondary lymphoid tissues. Primary, what do you have to think? Only the stuff here that is in red, bone marrow and thymus. Everything else is secondary. Okay, so this is important slide showing that your primary is thymus and bone marrow. And look at that, it is red, it is bold, it is underlined, and yeah, that's important. Okay? Secondary, everything else. So as long as you memorize the primary, you know everything else is secondary in these tissues. Okay. All right, so now we're going to start to get into the nitty-gritty of how do you recognize your pathogen. Remember, you have those recognition receptors, right? Pathogen recognition receptors to recognize pathogens. Okay, so that was easy. Okay, so here, how is it that you're going to respond? Again, this is again very much redundant, we have seen before. I should take that one before. Let me see. I already talked about this, so you can go back and study based on that. Okay, what is important to know in this slide here? If you look on the surface of your pathogens, you're going to find those little things sticking out. Those are the proteins, okay? Now, if you look here at this, let's say this guy is a bacterium, okay? It has a lot of those little things sticking out. It's not one or two. And usually those are some types of carbohydrates or some type of protein. Let's say it's carbohydrates on the surface, okay? Now, the way that your cells recognize a pathogen-associated pattern is not because these molecules are only present in pathogens. That's not the case. The same molecule can also be present in one of your own cells, okay? But your immune system knows not to attack your cells. And now the question is, how does it know that? Okay? The answer is that the number of these things sticking out in the pathogen, that number is much higher than what you would find in a cell. So let's draw a cell here, okay, with little green things sticking out. I meant green. Okay. Little green things sticking out. So here is something here, and it's exactly the same. I'm trying to put it as the bacterium. Okay. If your cells have the very same type of molecules that you're going to find over there, so how are you going to know which one to attack and which one not to attack? It has to be in the density of these patterns. Your own cells do not have as many of those molecules as what you're going to find in the surface of a pathogen. Okay? So the pattern, when you see a lot of these patterns, is concentrated here. And that is the trigger for recognizing. Yes, I'm blowing the time again. All right. So we're going to stop in this slide and think about pattern recognition. I'm going to come back on Tuesday.